Hi guys, so here let me share screen here. That's this one here. Okay. So today we are going to talk about cell division and the DNA replication. We will split it into two portions. The first portion is to talk about the cell division, and then the next one we will talk about the DNA replication. So in this lecture, we will cover different area, including cell cycle. Cell has different stages. So we want to learn about the cell while it's single cell, but during the time it will grow, and during the time it will start to divide, and then during the time it will be divided. Or there will be the situation that cell will never get divided, like cell then differentiate into specific type, like muscle cell, nerve cell, those fully differentiate. Or you may learn that the white blood cell, for example, those fully differentiate cells will not go through the cell division. So that is a stage of the cell. Then we will learn about two types of the cell division. One is called mitosis. The other one is called the meiosis. And then because when you have the cell division, you will basically, everything in the cell will be divided into two. And the most important part of that division is the chromosome or the DNA, the genetic code. So before you divide them, the cell need to be able to replicate that contents. So that DNA replication is important. So this is a typical cell, the human cell. In this lecture, we will focus on human cells. So you can see that cells has a nucleus contain the DNA. You can also see different organelles, and if you're interested, I have another lecture talking about the cell physiology. You can look into to learn different organelles. But basically here you get the idea that these organelles eventually will be divided to become two after cell division. The thing that we focus on is the DNA, because the nucleus contains the DNA contains the genetic codes to serve the function of the cell. We got this genetic code from mother and the father. So in a somatic cell, like typical cells, body cells, we will have a pairs of DNA sequence or DNA sets. One set come from mother, the other set come from father. So we have two sets. Each set has 23 chromosomes. So a total body cells would have 46 chromosomes. So this is the DNA packaged in one chromosome. So DNA, later you will see that here is a picture showing you that this is a DNA sequence. This is genetic code, GCAT. We have a double strand, so one strand and two strand form as a helix. And this string will then, it's very long, so we need to coil it, package it around these. This is called these are the proteins. These are the histone proteins. And then it will wrap as a chromosome. Then this chromosome will be in the nucleus. So that is DNA, the relationship between DNA and the chromosome. So a chromosome contains DNA coding as well as histones. Histones are proteins. So these two are different kind of molecules. Then we have this chromosome. We have in the somatic cell, like body cells, we have 46 chromosome or 23 pairs of the chromosome of these. So these are the example of these 23 pairs of the chromosome in the somatic cell or body cells. You can see that up to 22 and 23. So for a male, you will have one X and one Y. For the female, you will have two X chromosomes. So this is body cell, and we call it a 23 pair. We also simply put it to N. N basically is the number, N as a number, the variety of the chromosome, N types or N different kind or then N different chromosome. So basically we have 23 different chromosomes, and each one will have one from dad, one from mom. So the somatic cell we call it 2N because we have 23 as a number times 2N. So not times 2, yeah, 2N. So that is the somatic cells. We call these pair as the homologous chromosome. They are homology, they are similar to each other, they are different. Your mom look different from your dad, right? And so even though your mom has the hair gene, your dad has hair gene, but the color of the hair could be different. The height could be different. So the coding can be vary, but they are comparable. They are homologous to each other. So these pair are called homologous chromosomes. We don't call these X to Y as homologous because they are different. We call this homologous though, XX as a homologous. We usually call XY as sex chromosomes. Now, so the cell cycle, cell can have different stages. Let me show you this figure here. So cell in the very beginning, so this is cell division. You generate two new cells. Once you have these cells, say this is one, this one represent a set from mom, this set from dad. So you have 2N as the amount of the chromosome in a somatic cells. These cells will then go through the cell cycles. It will, in G1, G refers to a gap, meaning that the gap before division again. G1, G2 here. G1, the cell growing. So the small cell, newly formed baby cells will become bigger and bigger. Why is that bigger? Because you need to produce a lot of things. You need to produce proteins. You need to divide your endoplasmic reticulum. You need to divide your mitochondria. You need to have more and more of these organelles in a cell. So cell become bigger and bigger, and then it will have enough resources for division. So the first stage, G1, is a growing phase. So that's G1. And then it will go through when the cell is mature enough, large enough in a way, size is large enough, it will go through the stage called S. This stage is a stage that the chromosome will replicate. So you have 43 chromosome, it will replicate, or you call it duplicate. So you can see that here we have N, so it's 2N. Now 2N will times 2. Each one will generate an exactly identical copy of the chromosome. And then you will go through another gap. It's called G2. G2 is the preparation for cell division. And then we will go through cell division, and then new cell occur. New cell will go through this over and over again. So this is the typical cell cycle. These cell cycles apply to stem cells. Those are the cells can be divided, can reproduce themselves. However, there are cells that will not remain as a stem cell. They will be differentiated. When the cells are able to reproduce themselves, we call them proliferation. And one life becomes two life, right? Proliferation. But cells may jump out of this cycle to become fully differentiated cells. And for example, that the stem cell for the blood are the stem cell remain in this cycle in the bone marrow. And once there is a need of either whether white blood cell or red blood cells, these cells will start to differentiate into white blood cells or red blood cells to serve its function. And so that in that stage, we call it G0. So a fully differentiated cell is a cell in the stage of G0. That the cells would not differentiate anymore. You cannot differentiate. You cannot, cannot be divided further anymore. You cannot get your white blood cells. The white blood cell has everything for a cell, but just itself cannot be divided further. You want new white blood cell, you go to the stem cells. Those are the stem cells in the bone marrow. Those will generate unlimited number of the stem cells, and then those stem cells can be differentiated into specific white blood cell. So that's the stage, different stage of the cell cycles. So G0 is the one that is a stage for a fully differentiated cell. So a quick question would be that in which stage you will see the fully differentiated leukocytes? That will be the G0. In which stage the chromosome start to replicate or finish its replication or complete its replication? That will be in the stage S. Yeah. Another thing I also want to mention is that this phase G1 as G2, this phase is also called interphase, because that is the phase before or between division. So that's the interphase G1 as G2. You need to know the sequence G1 as G2. Yeah. So the cell can be in the G1 and go to the S, or remain the G1 for a long time. The stem cell, sometimes stem cell remain the same amount because they don't need to duplicate. Like stem cell in the bone marrow, right? If they are not controlled, if their division is not controlled, then it will be very much like tumor. So it's very controlled. They are capable of division, cell division, but the division process is controlled. So they can remain in the G1 for a long time. And then if it's time's ready, they can go into the S phase. During the S phase, then 2n times 2, the chromosome number replicated. And then S will go into the G2 and then conduct this cell division. The stem cell in G1 may start to become differentiated cell going to the G0. So that's interphase. In the S phase, this is the time that we will only briefly talk about the DNA replication. In the next lecture, we will talk about more detail as how these chromosomes get replicated. So in the S phase, you have, we just show you two chromosomes represent two sets of the chromosome. One set from the mother, one set from the father. So you have 2n. Each chromosome, this part is called the centromere. that is the center. There is some protein wrapping around these chromosomes. So this is a chromosome, right? So this is a chromosome. So this is a chromosome. So very basic stuff is that DNA and the histone. And so basically it's just a stick in a way, just a stick in a way. And so we sometimes see it like this. So it's basically just a stick, yeah, wrap it. And but there is a portion we call it centromere. So in this centromere, is the packing is much tighter. So you see it very much like a vest. So it's narrower in that portion. So you can see that this centromere is not always in the middle of the chromosome. This one is the middle, but sometimes we have a shorter arm in part in one side and the longer arm the other. Sometimes this shorter arm is very short. Yeah, so there are different structure, different type of the chromosome. All right, so the during the S phase, each chromosome will make the same copy of that one. When they make the exact same copy of that one, we call these two sister chromatids. These two sister chromatids are identical to each other. You must know that your two sets of chromosomes, one from mom and one from dad, are not identical because your mom and dad are not identical twins, right? So their chromosomes are not identical. But from the chromosome, one chromosome, it replicates to a sister chromatid. These two will be identical because that is exact copy. Exact copy. It doesn't really know what is the alternative. Basically, the only copy they can make is from their sister chromatids. So it will be making another set, exact the same one. So we have this. And then we will have 46 times 2 chromosome after the S phase. So you will see that if you look into the microscope, you will see that after the S phase or beginning into the mitosis, we call it prophase, the nucleus become very highly condensed because it has a lot of stuff. Yeah, so that's that. So the key here is the sister chromatids are produced after chromosome replication. These sister chromatids are connected in the centromere. So that's something. So it's not, the reason is that so you make this replication, you want to organize it, right? And so you tighten it. These two will be tightened together. So that's the space. So then it will go into the SG2, then it go into the cell division. Cell division has two types. The body cells, body stem cells, will produce exact same body stem cell. Stem cell in the bone marrow will produce another stem cell in the bone marrow. One cell will become two, two will become four. So that is the mitosis. That division, we call it mitosis. So here is one. If we don't look at anything between, these will produce two, and these two are identical to each other as well as identical to the mother cells. So that is the goal. That is a result of mitosis. You have one cell, you produce two identical cells. Okay, so that's that. Then how do we do it? First, during the S phase, each chromosome replicate and connect at the centromere. Then you will pull these sister chromatids away. You got a copy, right? They will be aligned And then I don't think I have the picture about that one there, but they will be aligned, and then these, each copy will be separated. And because you make the identical copy, you have 23 chromosome, 46 chromosome, 20N, 23N, sorry, 2N, and then it will be divided. And you only need to divide it once because you make one copy and you split them into 2 cells. So that is the mitosis. So the mitosis can be divided into different phases, 4 phases in a way. We call them prophase, metaphase, anaphase, and a telophase. So in this one, show you these 4 different stages during mitosis. So before the mitosis, we have the interphase. This interphase include the G1, S, G2. S is the stage that we have DNA replication. Then the prophase is the one, then basically during the prophase, your nucleus membrane become dissolved. Then you have the metaphase, that these sister chromatids are aligned in the equator of the cell, and then it will get pulled. The centromere will get pulled by the spindles apparatus, and then it will be pulled into 2 different ends of the cell, and then it get divided. So that is the mitosis. And so the key here is that the mitosis, you generate identical daughter cells. Another thing about the cell division is the meiosis. So mitosis is the mechanism that works very well for the somatic stem cells. You have the stem cells in the bone marrow, it will produce exact the same stem cells in the bone marrow. However, in the sex cells, in the male is the sperm, in the female is the egg or oocytes. The thing about those cells is that we don't want to have the exact, we don't want to have the full number of the chromosome, right? For example, the somatic cells, like this one here, you have 23 pairs. Do I have another picture? All right. All right. For example, just like this one here, you have 23 pairs of the chromosome. But you don't want that in your egg cell or your sperm cell, because your sperm will merge with an egg, and then that complete a cell. So when you generate the sex cells, you want to have the chromosome number to be half. You don't want to have 2. You don't want to have the diploid cell. You want to have an haploid, meaning half. Not di means 2. D or di means 2. You don't want to have 2 for each one. You want to just have 1 for each one. And so you can emerge the other cells to make it complete. And for the production of these sex cells, mitosis is not the one that we will do. We basically will still divide it, but we need to divide it further. We don't want to divide it just, we would replicate it, but we will divide it 2 times in order to make the chromosome number to be only half from the stem cells. So that division is called the meiosis. So that is meiosis. So meiosis is unique process to produce half number chromosome in the sex cells. These sex cells, we also call them gametes. Collectively, that's the eggs and the sperm cells. There will be only 23 chromosomes. We call it 1N, not 2N, or we call it haploid cells. So that is the meiosis. So how does meiosis do? So meiosis basically goes through two divisions. So why is that? Because, for example, in the mitosis, you have one cell, you divided it one time. You have one cell, you divide it only here one time. But for the meiosis, you have one cell divided, and then it's not stopping here. It will go further division. So it gets divided twice. So meiosis get divided twice. So that's one thing. And then by doing so, you have 2N diploid, then you will, in the end, your daughter cell will be only 1N. So that's that. Another thing about the meiosis is the sequence. So first is that you replicate it, right? During the S phase. So you have sister chromatids connect with the centromere. If it's the mitosis, then the sister chromatids will be separated, just like this one here. The sister chromatids will be separated, or like this one here. These sister chromatids are connected with centromere, they will be separated. But in the meiosis, during the first stage, it's not the sister chromatids get separated. It's the homologous. Your father chromosome will be separated from your mother chromosome. You have 23 chromosomes. In that portion, you may have chromosome A, that's totally father, chromosome 1, totally father, chromosome 2, totally mother, chromosome 3, etc. But it will be separate based on the homologous pair, but not the sister chromatids. So these sister chromatids will not be divided during the first division. And then during the second division, this is time that sister chromatids will be separated. So that's a unique part. The sister chromatids will not get divided until the second stage of the meiosis. So that's it. So you have that. Here is the summary of this meiosis. Meiosis contains two stages. The first stage, the homologous pair will be divided, and then the second stage, the sister chromatids get divided. You will generate from a 2N during the S phase. 2N will times 2, but this 2N will be divided. So after the first stage, you have N times 2. You have only either mother or father, but you still have times 2, meaning that you have identical copy of each chromosome. That's the sister chromatids. And then during the second stage, these identical copy will be divided. So it will generate only N chromosomes in the daughter cell. So that's that. So each chromosome, each daughter cell, each gamete will have only 23 chromosomes, not 23 pairs of the chromosome. So that's meiosis. So a good summary between the mitosis and meiosis, that here the stem cells, we have the 2N, 1N, second N, one from father, one from mother. We have 2N, 23 pair. During the S phase, each chromosome replicate, sister chromatids connect with a centromere. Division is that these sister chromatids will be divided. So the result is these are the one cell that will be identical to this one, and these will be another cell, will be identical to this one. So the daughter cell will be identical to the mother cell. There will be no mother cell. The daughter cell will be identical to each other. So that's the mitosis. As for the meiosis, again, during the S phase, you replicate your chromosome. Sister chromatids connect with the centromere, but they go through two divisions. The first division is not to separate the sister chromatids. It's separate mother from father. Homologous pair will be separated. These are the homologous pair will be separated. And then during the second division, the sister chromatids are separated. So that's meiosis. All right, that's it. Before we end, I want to introduce one concept that is called the crossing over. So what is it? It's because that when we conduct this meiosis, these chromosomes are very near to each other. And then there will be situation like this. So these chromosomes are very near. Before they divide it, before they separate apart, these two strings will have the chance to get exchanged. So these homologous pair will get exchanged. So we will end up chromosome like this. And then if you divide it further, you have one daughter cell completely blue, one daughter cell largely blue, but a little bit red, one daughter cell largely red, but a little bit blue, one daughter cell completely red. So you will have four totally different daughter cells after the meiosis. We don't see the crossing over during the mitosis is because that during the mitosis, there's no chance, even though if these two crossing over because these two are identical, so they won't cause any difference. But the meiosis is different. Meiosis is that these pair will align with each other, and the distal portion, especially these distal portion, are swinging around. They are not tight. The only thing got tightened is the centromere. So this end part will swing around, and then they will be crossing over, got exchanged. So that's that. So this one summarizes the two types of cell division, mitosis and meiosis. Mitosis conducts one division, meiosis conducts two division. Mitosis is the division used for the somatic cells, the body cells. Meiosis is a division for the gametes, for the sex cell. So the outcome, the result of this meiosis is the reduction of the number of the chromosome. Sperm cell don't have the full number of the chromosome, it only has half. And that's why sperm meets egg will make it full. Crossing over increase the diversity of the daughter cell. Crossing over only happens during the meiosis, meaning that the gametes will be very different from each other. One of the reasons is the crossing over. In the meiosis, the first division is homologous division. The sister chromatids are separated in the second stage. And during the meiosis, that the daughter cells are totally different from each other, but after the mitosis, the daughter cells are identical to each other. So that's that.